Module 1, a review of protein synthesis. So now you know that proteins are synthesized via the ribosomes. Well, let's take a closer look at how proteins are made. The building blocks of proteins are amino acids, and the types of amino acids, their order, and how many of them are joined together determine what protein is made along with the properties of that protein. Some proteins are a part of a group of chemicals called enzymes, and enzymes behave as catalysts. A catalyst helps a chemical reaction go more quickly, yet the catalyst is not used up by the reaction. Some proteins act as hormones. Some are antibodies fighting infections. Others make up larger components of your body. There are literally thousands of types of proteins needed for living processes. This is why it's important to learn about how they are made. Protein synthesis in the cell involves two major steps. The first is called transcription. The root word script is the same root we get the word scribe from. A scribe copies down information. Well, in transcription, a portion of the DNA in the nucleus unwinds so that it can be copied. The copy that results from this transcribing is called messenger RNA, or mRNA. It travels out of the nucleus to the ribosomes. There are specific amino acid codes on the mRNA that call up the correct amino acids. These codes are called codons. And this begins the process of translation, translating the DNA information to amino acid information. Each of the amino acids is attached to a short strand of transfer RNA, or tRNA. tRNA has a special anticodon sequence that matches up with the specific codons on the mRNA. When they come together, the amino acids that were attached to their respective tRNAs will chemically bond together, creating the protein strand.
There's a great analogy in your text that better describes how transcription and translation work. Suppose I want to make a cake from an old-fashioned, authentic recipe. Well, I don't have the recipe and I can't find it on the internet, so I have to come to the library to find it. I find the recipe in one of their reference books, like this one. Well, I can't check it out, so I make a copy of the recipe onto the recipe card. Then I take the recipe card to my kitchen, where I gather the ingredients and make the cake. The reference book in the library is like a cell's DNA in its nucleus. I found that portion of the information I needed to make a copy. My recipe card then is the messenger RNA. I take this information to my kitchen, which would represent the ribosome of the cell, where all the components are waiting for me to assemble them correctly. Those components would represent the transfer RNA. So transcription occurred at the library, or nucleus, and translation occurred in the kitchen, or ribosome. The result? A protein, or a delicious slice of cake.